So, biological membrane. Maybe I should turn off the light. Would that be better? You can all hear me okay? Whoa, biological membranes. So, what is the main component? If we look at this molecule here, what is the main component of a biological membrane? Anyway, what's the name of this molecule here? Phospholipid, right? The phospholipid biological membrane is two layers of phospholipids. Now, so if we look at this molecule, why do we call it a phospholipid? What's this part of the molecule? It's a phosphate, right? It's a polar group. And the polar group is what? Hydrophobic or hydrophilic? Hydrophilic, right? So the phosphate head is always going to be facing either the cytosol or whatever kind of media that the cell is present in, or any kind of extracellular fluid. Okay? Now, if this is hydrophilic, then this part is what? Hydrophobic, right? These are the carbon tails that are present on the molecule. And they're hydrophobic, so they face the inside. So, what's the point of all this? Well, most of your cells are made of this, right? Most of your organelles are made of phospholipid bimembranes, biological membranes. And what's the challenge we have? Well, to be alive, things have to be able to pass through this biological membrane in one way or the other. So what we have here, this diagram, we call it the fluid mosaic model. Is anyone familiar with that term? Did you use it in basic cell? I think it's pretty basic. So, fluid mosaic model, it means that these lipids can sort of move throughout the membrane. It also means these proteins are usually able to move throughout the membrane. For those of you who've read my thesis, you have to write a thesis if you're a doctor. We also found that the lipids can sometimes flip from one side to the other of a biological membrane. So they're actually very dynamic. Don't just think of it as a wall. It's literally a fluid mosaic. Things can move around in there, and they all have a function. Sometimes the movement of lipid from one side of the membrane to the other might lead to division in organelles, right? I mean, I'm up here, I'm alive and well. You can imagine that this thing can do a lot, right? So, what's the problem we have with biological membranes? As I said, things need to pass through this biological membrane in one way or another. Now, we're talking about molecules of different sizes. Am I talking too fast, too slow? Good. Yeah, molecules need to move past. There's small molecules, big molecules, charged molecules, uncharged molecules. So things like gas, oxygen gas, these can pass through the membrane pretty easily. CO2, nitrogen, benzene, these can just diffuse across the membrane. Did anyone have an idea why these things can diffuse so easily? No? Well, I'm sure you do, but that's a long question. So, they're hydrophobic. What we're going to see later on is there's certain channels. So as long as things are able to if they have an affinity for the hydrophobic part of the membrane, somehow, either because of a channel or because of their inherent properties, they can stay there long enough to be passed through. All right? Did everyone follow that? I'll clarify a little more later on. So gases, okay, small hydrophobic molecules. They also have small uncharged polar molecules. Now when you guys see this slide, they're probably going to put water on there. Water doesn't really diffuse across the membrane. I think this slide is probably old, 1995. We're going to see later on how water actually passes through the membrane. And it does diffuse a little bit, but for all practical purposes, water does not diffuse through the membrane. So when you see that on your slide, either he's going to put this up or you'll just make sure you cross it out. Things like urea and glycerol, these are also able to diffuse through the membrane. Can anyone give me another example of a molecule that diffuses through the membrane? Some of you guys were probably drinking stuff that diffuses through biological membranes this weekend. From the looks of it, those of you who are hungover, why'd you get drunk? Because the brain-blood barrier is permeable to ethanol, right? So when you drink alcohol, the ethanol just slips through there, makes you do all kinds of embarrassing things, and come to class hungover. So that's one molecule. Things like sugar. So obviously we need sugar to survive. Large carbon molecules, sugars, fats. These can't pass through the membrane, right? They're too big, they're uncharged. So what we're going to see later on is what kind of channels will be able to allow this to pass through the membrane. And ions. What are ions? So charged atoms are not able to pass through the membrane. They're just going to bounce right off. Here's some examples of the more important ions that function in the human body. Okay, any questions about that? Is this review? Have you guys seen all this before? Yeah? Okay. Here's another diagram, probably going to appear in the PowerPoint presentation, just to show you the variety of different molecules that can actually appear in a biological membrane. Sugars, proteins, so you can have proteins that go through the membrane, and this is what we're going to be looking at mainly, channels that are allowing molecules, sugars, ions, to actually pass through this membrane. Clicker's not working anymore. Okay. So there are different types of transports. We're gonna look at the different kind of transport across the membrane. You can divide them into two large categories. There's going to be passive transport and active transport. Did anyone read the book? Do you guys read the book before the lecture? After? Who's already read the chapters, the readings? All right, 2 people. So, passive and active. Can anyone tell me what the difference is between passive transport and active transport? Yes?
Active requires ATP.
Active requires ATP. Isn't necessarily ATP.
Well, no,
ATP, GTP, is there anything else? Okay, so what I'm trying to get at, it doesn't necessarily have to be coupled to the hydrolysis of a phosphate. Okay? When we talk about active transport, we're talking about reactions that require energy. Anything that requires energy. So if an ion, generally speaking, an ion is going to move, how do ions move across the membrane? Anyone? So channels, but what I was going at is energetically, how does this movement occur?
down the gradient.
Down a concentration gradient, okay? It's going to move from high concentration to low concentration, okay? This is an energetically favorable reaction. We call this passive movement across the membrane, all right? Down a concentration gradient. Now, what was mentioned before is that active transport requires energy. So the movement of a molecule down its gradient is an energetically favorable reaction. Therefore, we could also couple this to a reaction that requires energy. So there are some transporters that they mentioned in the book. We call them, what kind of porters? Antiporters and symporters, right? So antiporters and symporters are examples of transporters that are going to use the energetically favorable movement of a molecule, an ion, down its concentration gradient to power an energetically unfavorable reaction. For example, the movement of a molecule from higher concentration to lower concentration. Did everyone follow that? It's an important point, but I think you get it. So again, so there's passive transport, generally speaking, the movement of a molecule down its concentration gradient, and there's active transport. Active transport is the movement of a molecule in an unfavorable direction, therefore it needs to be coupled to an energetically favorable reaction, such as the hydrolysis of ATP, GTP, or the movement of passive ions. All right? So what are the different kinds of channels? There can be non-mediated passive transport, which means that a molecule is going to diffuse across the membrane. Okay? So we talked about those. Gases can diffuse across the membrane, like oxygen, what else did we talk about? Benzene, things like that. Or there can be a channel, as we discussed before. So if a molecule is too small, it can still move down its concentration gradient if you just poke a hole into the membrane. That seems fairly obvious. Okay, we can add transport-mediated passive transport. This means that there is a channel, but there's probably something that opens or closes. In other words, it changes conformation based on some kind of stimuli. We'll see examples later on. That's going to allow the passage of something down its gradient, right? Does everyone follow that? We don't necessarily want things moving through our channels all the time. Sometimes we close them, sometimes we open them. Really honest. And then, of course, we have channels that are powered by ATP that are going to allow the movement of particular molecules. Okay? Round it up. So there are examples. Oxygen, which can diffuse across the membrane. Sodium, which needs a channel to pass through. We have glucose. Now, what's an example of a transporter that's going to move glucose? I think in the book they talk about uniporters, right? So a uniporter can move glucose downstream. And something you guys are going to be looking at a lot is the Na+, K+ pumps. Just out of curiosity,